Hello students, welcome to EPG Pathshala. Today we are dealing with the molecular biology of the nematodes. Starting with the learning objectives, to study the general biology of nematodes, nematode life cycle and door formation, adaptations and habitats of parasitic life and a comparative account of molecular biology of these species, studies on nematode genome analysis and interpretation, molecular tools for understanding biology of nematodes, studying popular model organisms and their constituent properties and characters, ecology and evolutionary studies on nematode species. In this module, we will be dealing with the general biology of free living nematodes, the most common example being the Caenorhabditis elegans. We are studying the molecular biology of this free living nematode, focusing on the life cycle, how it is starting from egg to egg and completing within 3 days, starting with the larval development, how it progresses with larva 1, 2, 3, 4, or it goes on to the dauer stage where the arrest stage occurs. And we are also dealing with the genome analysis. It is the first multicellular organism where this entire sequence of the genome was first studied. It is a very interesting model system and we will be covering the various aspects that are of huge interest for learning genome biology. We are also focusing on the adaptations of the organism and the evolutionary aspects. There are so many conserved regions found in this organism. So, there are regions which are showing similarity to other organisms like humans. There is a 40% similarity which the C. elegans genomes shows with human disease conditions. Another very beautiful aspect of the C. elegans is that this organism is entirely transparent. This gives us a window to look into the cell fates, starting from the zygote, how each cell is developing and how it forms different organs. So, it is a beautiful model organism to be able to follow the cell fates of each cell, giving us the complete cellular lineage and helps us in understanding the development better. Free living nematodes. The nematode Caenorhabditis elegans was proposed as a genetic model for gaining an insight into concepts of developmental biology and neurobiology by Sidney Brenner. The word Caeno means recent, Rhabditis refers to rod and elegans refers to nice. C. elegans is a non-parasitic free living nematode found in the soil. The unique features of this organism that make it an outstanding experimental model system includes its small size, rapid life cycle, transparent body and well annotated genome. Other features include large brood size, ease of cultivation and maintenance, long-term cryopreservation, quick regeneration time, invariant cell number and development, and the ability to reduce gene activity using RNAi. In addition, the fact that these organisms cannot grow at body temperatures makes them quite benign to humans. A side view of an adult hermaphrodite worm Caenorhabditis elegans is shown in this diagram. The C. elegans has a simple anatomy with a small number of tissues and internal organs such as mouth, pharynx, intestine, gonads and collagenous cuticle. The various parts can be clearly observed in the diagram which shows pharynx, oocyte, intestine, uterus, vulva, eggs, body wall, gonad and the anus labeled from anterior to the posterior end. Parasitic nematodes. Parasitism is believed to represent a derived character from an evolutionary perspective. This implies that all parasitic life derives from free living ancestors. There are certain characteristic preconditions for a parasite to be: first, identification and contact with the host in a specific manner. Second, survival and propagation inside or on that host. Third, ability to derive food, and fourth, capable of successful reproduction in this new environment. These pre-adaptations are a set of characters that explain the fitness benefit that is assumed for the parasite to be to begin host exploitation. This pie chart shows parasitic infections, how they occur and how many people really are hit by these infections. The pie chart clearly shows that the intestinal roundworms are affecting the major chunk of population, reaching to around 1.4 billion. Around 300 million of the population is affected by malaria, and the other parasitic infections which are also causing great extent of harm include dracunculiasis, African trypanosomiasis, leishmaniasis, Chagas disease, foodborne trematode infections, amoebiasis, lymphatic filariasis and schistosomiasis. The following diagram depicts the two sexes present in the C. elegans. The first is a self-fertilizing hermaphrodite depicted by XX, and the second is a male depicted by X0. In the hermaphrodite, the various parts labeled in the diagram are ovaries, oviducts, spermatheca and a single uterus, while in the male we have a single lobed gonad, a vas deferens and a tail specialized for mating. Biology of C. elegans. C. elegans is unsegmented, vermiform and bilaterally symmetrical with a cuticle integument and fluid filled pseudocoelomate cavity. C. elegans has two sexes, hermaphrodites and males. Most individuals are hermaphrodites, males comprising only 0.05% of the total population on an average. The males have a single lobed gland, a vas deferens and a tail specialized for mating, whereas the hermaphrodites have two ovaries, oviducts, spermatheca and a single uterus. The eggs are fertilized internally by sperm produced by the hermaphrodite or by sperm contributed by a male. Eggs are laid by the hermaphrodites and development is completed externally. C. elegans has 5 pair of autosomes and 1 pair of sex chromosomes. At 20 degrees Celsius, the average lifespan is approximately 2 to 3 weeks with a generation time of approximately 4 days. After hatching, which is almost 14 hours after fertilization, they pass through 4 larval stages labeled as L1 to L4. Hermaphrodites produce all their sperm in the L4 stage and then switch over to producing oocytes. During unfavorable situations like overcrowding or absence of food, C. elegans can enter an alternative third larval stage called the Dewar stage or the arrest stage. When self-inseminated, the wild type worm may lay about 300 eggs. However, on insemination by a male, the number of eggs may exceed 1000. Hermaphrodites which self-fertilize produce only hermaphrodites. When hermaphrodites mate with males, 50% of the progeny is male and 50% hermaphrodites. A short history of C. elegans research. C. elegans was developed by Sidney Brenner in the year 1963. Mutants were established by Brenner in 1974. Post-embryonic cell lineages were determined by Sulston and Horwitz in 1976. Programmed cell death was first identified by Horwitz in 1982. Complete embryonic cell lineages were determined by Depi et al. In 1983. Complete connectivity of nervous system was established in the year 1986. RNAi and miRNA was discovered in these worms in 1991 to 98, for which Fire and Mello won the Nobel Prize in 2006. First use of GFP or the green fluorescent protein in animals was depicted in the year 1994 by Chalfie, for which he also got a Nobel Prize in the year 2008. The first multicellular animal whose entire genome was sequenced was the C. elegans. Life cycle of a typical nematode is depicted in this flow chart. The adult is capable of egg laying, and then in utero development begins, which starts with cleavages. The eggs are laid at the gastrula stage, approximately 30 cell stage. Then begins the ex utero development, which proceeds for almost 9 hours, showing the 1.5, 2 and 3 fold embryo. Then this is followed by the larval stages, the L1 to L4 stage. The L1 molds into L2, which molds into L3 and which further molds into L4. In case of crowding and starvation or unfavorable conditions, the L1 larva undergoes a rest stage and enters the Dwyer formation. The L4 stage otherwise becomes the young adult, and the cycle is completed within almost 3 days. Dwyer larva. Developmentally arrested alternative third stage larva is the Dwyer larva. It forms in response to high population density, high temperature and low availability of food. It is a non-feeding stage where the buccal cavity is sealed and it survives on stored food. This larva can live for up to 70 days. Post-Dwyer adults have normal lifespan and continue in the life cycle. Now we will understand the molecular basis of Dwyer formation. In C. elegans, Dwyer formation is induced by elevated temperature, shortage of food and high population density. Genetic analysis has shown Dwyer formation defective mutants and Dwyer formation constitutive mutants, which are the two forms of genetic mutants. The DAF-D or the defective mutants do enter Dwyer stage even under unfavorable conditions, whereas the DAF-C or the constitutive mutants form Dwyer larvae even in the abundance of food. C. elegans population density is sensed by pheromonal cues indicating overcrowding, and a group of glycosides called arachosides constitutes the Dwyer pheromone. The regulation of Dwyer formation includes negative regulation of the nuclear hormone receptor DAF12, which in turn is guided by environmental cues that act by repressing a steroid hormone. Now coming to the genome of C. elegans. C. elegans was the first multicellular organism to have its genome completely sequenced. Sequence was published in the year 1998, although a number of gaps were present. The last gap was finished by October 2002. The genome was sequenced using the clone by clone approach. The genome size is 9.7 into 10 raised to 7 base pairs or 97 megabases. There are some large intergenic regions containing repetitive DNA sequences. Introns comprise 26% of the genome. Around 35% of C. elegans genes have human orthologs, showing a lot of promise for development studies. Remarkably, it has been shown repeatedly that human genes replace their C. elegans homologs when introduced into C. elegans. Similarly, many C. elegans genes can function similarly to mammalian genes. There are some large intergenic regions containing repetitive DNA sequence. Many genes are arranged in operons. C. elegans and some other nematodes are the only eukaryotes currently to have operons. Now dealing with genetics of the C. elegans. C. elegans makes a very efficient model system due to ease of genetic manipulation. Researchers screen for mutations anywhere in the genome directly in a single mutagenesis experiment and then go for linkage determination. The characteristic nature of self-fertilization allows maintenance of mutant alleles except dominant lethals by self-propagation post-mutagenesis of hermaphrodite worms in F1 or F2 progeny without mating, making the process of obtaining their progeny easy. The C. elegans has a remarkably short 3-day life cycle. This implies that 2 generations of the mutant homozygotes can be identified almost 1 week post mutagenesis. The method of freezing and recovering C. elegans provides an added advantage of preserving mutant strains, avoiding any chances of acquiring unwanted suppressors, modifiers, or additional background mutations, or losing important mutations. The traditional use of genetics in C. elegans deals with forward genetics, which starts with the screen or selection to identify mutants with a particular phenotype, after which the interference of the wild type role of the gene is gathered from the nature of the mutant phenotype. It is very interesting to know that the Nobel Prize in Physiology or Medicine was awarded in the year 2002 for C. elegans research. The Nobel Prize in Physiology or Medicine 2002 was awarded jointly to Sidney Brenner, H. Robert Horwitz and John E. Sulston for their discoveries concerning genetic regulation of organ development and programmed cell death. C. elegans is a beautiful model system. Let us now study some advantages of C. elegans as a model organism. It is simple and easy to grow. It has a short life cycle. Powerful genetic studies can be conducted because it is both self-fertile and cross-fertile. It is transparent and has a small size. It has an invariant cell lineage. Fully described anatomy and development are present today. The genome is completely sequenced. Tools like RNAi knockdowns can be performed easily, providing a molecular handle for researchers. Stocks can be frozen and preserved. No expensive animal house costs and easy maintenance. They do not present any biohazard and they have a large size progeny. Moving on to cell fates in C. elegans. C. elegans is characterized by simplicity of physical as well as genomic design. The development from a fertilized egg to an adult worm occurs in a manner similar to that in humans. Development begins with a single cell, the fertilized egg, which through consequent cellular divisions gives rise to a small worm inside the eggshell. Prior to hatching, growth and sexual maturation of the young worm occurs by cell division as it undergoes molting and passes through 4 successive larval stages. The hermaphrodite worm produces its own eggs soon after the final molt to the adult stage occurs. The entire developmental sequence from egg to egg is completed in only 3 days. Direct observation of the developmental stages from the single cell egg to the multicellular adult was done to map the cell lineages of all the cells. A particular precursor pursues the same pattern of cell divisions in each individual. Therefore, with very few exceptions of the fate of very descendant cell can be predicted from its position in the lineage tree. The outcome in the nematode is almost perfectly predictable simply because the pattern of cell to cell interactions is highly reproducible and is accurately correlated with the sequence of cell divisions. This diagram depicts the lineage tree for the cells that form the gut or the intestine in C. elegans. Although the intestinal cells from a single clone, the cells of most other tissues do not. The anatomy of this worm has been described in extraordinary detail, and mapping out of the exact lineage of each cell is possible. The genetic control of development of this multicellular organism has been traced, and it is a beautiful organism because of the transparent body. One can really observe the fate of each cell proceeding from zygote to the adult stage. Proceeding to the ecology and evolution of the C. elegans. The genus Caenorhabditis belongs to the order Rhaptida, which is a part of the class Chromidoria. Caenorhabditis is included in the phylum Nematoda, which in turn belongs to a larger group of the clade Ecdysoa. The Ecdysoa is characterized by organisms that shed their cuticle by molting or ecdysis. Although Caenorhabditis species are free living universally, the Rhabditae does include parasites of animal and plant species along with free living species inhabiting a variety of terrestrial and aquatic ecosystems. C. elegans provides a rich base for comparative evolutionary analysis among different nematode species. Evolutionary studies throw light on a wide range of biological processes like regulatory pathways and organogenesis in related species. Further studying the Caenorhabditis species in the animal kingdom, the phylogenetic tree A shows placement of the Caenorhabditis species in red among the metazoans based on sequence data from 2 ribosomal subunits, 8 protein coding genes and mitochondrial genomes. The phylogenetic tree B places the C. elegans in red among the named Caenorhabditis species grown in the laboratory. Species in red have hermaphrodites and males, whereas species in blue have females and males. Coming to the evolution orthologs. The experimental flexibility and similarities in the cellular and molecular processes such as metabolism, organelle structure and function, gene regulation, protein biology, etc. of C. elegans and other related organisms in the evolutionary tree make it a very successful model organism for understanding biological processes. Around 38% of protein coding genes present in C. elegans are reported to have predicted orthologs in the human genome. Although 60 to 80% of human genes have an ortholog in the C. elegans. Further, 40% of the genes associated with human diseases are known to have clear orthologs in the C. elegans genome. Therefore, discoveries in C. elegans would have significance to the study of human health and disease and seem to be very promising. Let us now summarize what we have learnt in this module. Sidney Brenner opened gates of understanding of various aspects of biology by understanding the Caenorhabditis worm. A vast array of biological analysis has been conducted in the C. elegans worm, where development analysis has been undertaken and cell lineage studies have been performed, which have provided a real-time assessment of development of cells starting from a single cell stage leading to the complete development into an adult. and it is really aided by the transparent nature of the worm. The sequence of this worm has been completely sequenced and the genome is completely annotated. It is one of the first multicellular organisms where this has been done, and it has provided a great molecular handle for us to be able to compare genomes across animals. Comparative genomic analysis has also revealed that the HGT or the horizontal gene transfer has been a fundamental process by which genetic acquisition of various genes has been conserved across species. Extensive gene family expansions and horizontal gene transfers are considered as two modes by which the diversification of the nematode genome has occurred. Thousands of conserved genes have been found in the genome of the C. elegans. These conserved genes are found in different animals including humans. It proves to be a very promising study because around 40% of the disease causing genes in humans have clear orthologs in the C. elegans. Therefore, various diseases can be treated and it is a very promising model. Thank you.